So we'll continue now with chapter 4, which is on carbon nanotube-based nanonetworks. So this is yet another technology that could be used for nanonetworking. Actually, this technology is partly in use as of today. So there's already too much investigation going on in use of carbon nanotubes, but our focus will be mostly understanding how it could be used for networking purposes. But to understand it, of course, you first have to understand how the carbon nanotubes are formed and what are the properties of carbon nanotubes. So we'll be discussing first the nature of carbon nanotubes. That will be followed by the discussion of the comparison with microtubules, comparing carbon nanotubes against microtubules. Then we will be talking about using nanotubes for interconnects, and we'll be discussing motility assays in that sense. And then we'll be discussing nanotubes as field effect transistors. Next week, since we will have the exam, we will not have lecture, but in the following week, I will be adding more into this chapter, so this table of contents is not complete yet. So, carbon nanotubes, in short, we'll call them CNT. Most of the research on carbon nanotubes typically focuses on interconnecting them with computer chips. How we can make use of carbon nanotubes in the design of computer chips. Our interest is mostly in the ad hoc approaches in nanoscale networks. So we'll be looking at approaches where you have you don't have a precise layout, as in the case for the use of carbon nanotubes in chip design. Carbon nanotubes typically has some very interesting properties that are very useful, and that would be very useful for networks, but also for a variety of applications, like sensor coverage will benefit from finding better ways to communicate among even smaller sensing elements, and also the use of carbon nanotubes is in the design of nanosensors, designing sensors that can detect single atoms, for example. So sensor networks, wireless sensor networks research is slightly also shifting towards the design of nanosensors. As the development in the nanotechnologies continues, there is need for low-cost, robust, and reliable communication among these nanomachines. And the communication and signaling within these inorganic and biological or biohybrid nano systems of nano devices will allow us to deploy these devices in a very dense manner in a specific area, and you need to provide efficient communication between these distributed devices. We will focus on the inorganic carbon nanotubes in this chapter, but we will also talk about the similarities between the networks of these carbon nanotubes and the microtubular networks we have discussed in chapter 2. And much of the analysis of the carbon nanotube networks will typically apply to the nanoscale networks of microtubules as well. And we will see how we can actually use the two together. So you can imagine small nanotube networks with functionalized tubes sensing multiple targets inserted into a cell in vivo. In vivo means doing it in a live organism. So information from each nanotube sensor can be fused within the network rather than transporting it all the way to the destination. During the transport, you can do the fusion. And this is clearly distinct from the traditional and potentially less efficient approaches of using carbon nanotubes to construct the transistors for wireless devices that do the same thing. One of the most direct applications of these carbon nanotubes and nanoscale networks built on these carbon nanotubes would be the single carbon nanotube radio. We will be discussing that in the next lecture, not today. And although it has been promoted as a molecular radio, this single carbon nanotube radio, sometimes they refer to it as the molecular radio, it's actually limited to being just a radio receiver. However, it provides a very good example for showing the flexibility of the use of carbon nanotubes, and you're leveraging all aspects of these, all features of carbon nanotubes, typically for wireless reception of information at the molecular level. This design is actually very important. We'll later emphasize that once again. This design is very important because remember, when we say nanoscale networking, we always put the emphasis on the communication between the nanoscale devices. And that's important. We should do that. However, you should be able to somehow relay this information at the nanoscale to the human scale. And that's a difficult task. How can you design a device which will be able to both talk to the humans but also to the nanoscale devices? The design of this single carbon nanotube radio is important in that sense. It can serve as a gateway between the nanoscale and the human scale. So don't think of these carbon nanotube based nanonetworks as a different way, a separate way of designing nanonetworks. These could work hand in hand with the molecular communication systems we discussed in earlier chapters. You can, for example, do nanonetworking between cells using, for example, gap junctions or diffusion or whatever. That would work only in the nanoscale, and those molecules or molecular waves somehow arrive at these devices which also utilize the carbon nanotube radio to transfer the information at the nanoscale to the higher scale, which is where the human beings can do the communication. That means to the wireless devices we already know. Otherwise, if you cannot do it, the information will get stuck at the nanoscale and you cannot use it, which is useless. Actually, you will not be able to even detect that there was such a communication. So finally, manipulating a nanoscale network, both in terms of the construction of the network and also after you construct the network, maintaining that network will require the ability to manipulate the nanoscale components. Now, that is difficult because, as we discussed earlier, you don't have, actually we do have nanotweezers, but it is difficult to construct these nanoscale devices if you cannot hold and place that nano component besides the other to construct the larger nanoscale device. So nature has already enabled us with these biological systems which can do self-assembly of the components. That's very important because once you construct such an engineered cell, for example, it can reproduce and produce many of those nanoscale devices. But for the most part, we simply leverage the pre-assembled nanoscale components that were already in the cell. Like for the battery of the nano device, you don't try to come up with a nanoscale battery, but you make use of the existing mitochondria in the cell. As long as you provide ATP to that, it will work. A completely human engineered system from inorganic components will typically require a way of working at the nanoscale, reaching that nanoscale and doing that assembly process, which is pretty difficult. But not so surprisingly, most research up to now and most of the implementations have made use of this self-assembly feature to revert back to the biological approaches in the field. So, what are these carbon nanotubes? Carbon nanotubes are actually allotropes of carbon. An allotrope is allotropes are the elements that can exist in multiple forms. So, in each allotrope, the elements of the that elements atoms are bonded together in different ways. Each one would be a different allotrope of that molecule. So allotropes are different structural modifications of the same element. Okay? And the carbon nanotubes are formed in tubular molecules into tubular molecular structures, giving them desirable properties. So this is, as the name implies, actually a tube made out of carbon atoms. But we will see that we can organize them in different ways, and each one has a different feature. One important property of the carbon nanotubes is that they're very strong. They're bendable, but they're very strong. Another one is they can conduct electricity, and this is adjustable. You have different conductivity for different ways of forming carbon nanotubes. You can have very good conductors, as well as you can, for example, come up with semiconductors of carbon nanotubes. And also, they are very good thermal energy conductors. Again, this is also adjustable. You can adjust how much thermal conductivity it has. There are various ways of forming the carbon nanotubes. The simplest one would be the single-volt carbon nanotube. And you can have multiple carbon nanotubes that are inside each other.
Are
yes.
you able to construct these carbon nanotubes, or they are just naturally are constructed in the...
No, carbon nanotubes can be fabricated.
Fabricated by us?
Yes, by human beings. Yes.
Right.
We're not reusing what's already in the nature, but you're able to construct it yourself. Actually, the carbon nanotubes are known for their high aspect ratio. That means look at the width or thickness versus the length. That aspect ratio is very high, extremely high. In fact, higher than everything. Some have been observed with length to diameter ratios of up to 132,000 to 1. That means the thickness, consider the thickness of the carbon nanotube to be 1 nanometer. Typically we have it around 1 nanometer. The length may go as high as 132,000, 132 million. If you look at your book, you will see a much smaller number. That's the advance in technology. This is the more up-to-date value for that. The molecular structure of the carbon nanotube is significant, which affects actually everything from the strength of the carbon nanotube to conductivity of electricity and also thermal energy. And before actually going any further, let me explain it this way. Carbon nanotubes are actually formed from carbon nanoribbons. A carbon nanoribbon is actually a piece of graphene, where graphene is considered a regular hexagonal structure, where at every corner of the hexagon you have one carbon atom. And these hexagonal structures are aligned to each other, so that you get a single sheet of carbon atoms, nothing else, organized in hexagonal layout. This is graphene. Take a strip of it, that's a carbon nanoribbon. Okay? Can I get a piece of paper? Thank Thank you. Now, lay these carbon atoms as a single sheet. That will be, as I said, a carbon nanoribbon. Now, start rolling it. See, if you roll it this way, sorry, I'm not good in this. Of course, one layer. I can do it very well. This becomes a tube. Okay? That's a carbon nanotube made out of a carbon nanoribbon. Now, the important thing is how you roll it. Now, this is one way of rolling it. What I did was just roll it this way. I can also roll it from the corner with different angles. This is yet another carbon nanotube. Change the angle of rolling, everything changes. What changes is the strength of this tube, the electric conductivity, and the thermal conductivity, all depending on how you roll it. And that's what we will see now. Okay? So we will talk about something called a chiral vector. Okay, by the way, this is a single volt nanotube, because there is a single wall. You can make another roll around this, then it will be double. In general, we would call that multiple. Okay, let's focus on single volt. The rest is similar. Okay, so if you consider a single volt nanotube, we are going to identify what type of a single volt carbon nanotube this is according to its chiral vector, where the chiral vector is actually a two-tuple of n and m, where n and m are integer values, and these integer values are actually the factors of the vectors a1 and a2. And a1 and a2 are the unit vectors. Now let's look at this figure. This will explain it better. Okay? Now, see, a1 and a2. So this, the background is our graphene, or carbon nanoribbon at the moment. As you see, there's the hexagonal structure. And a1 and a2 are the unit vectors here. And by putting factors as n and m in front of a1 and a2 respectively, you define actually a vector. Different n and m values will correspond to different values. Like when m is 0, this line will be the chiral vector. When m is equal to n, in that case, this line will be the chiral vector. Okay? This vector t here is the axis of the tube. Like in my case, this is a tube axis, and the chiral vector now is showing how I'm rolling it actually. Okay? And some of these n m value pairs, the tuples, have special names. Like if m is equal to 0, it's called zigzag. If n is equal to m, it's called armchair. And the reason for that would be, if you take m equal to 0 and roll it that way, those hexagonal structures will end up with a zigzag shape here. If you roll it according to this n by n vector, where m is equal to n, then it's the armchair, because if you just look at this part and just rotate it 90 degrees, it looks like an armchair. The sitting place would be here if it's rotated, and these are where you rest your arms. That's why it's called an armchair, like what Tom says. Okay? So, according to how you wrap this ribbon, you get a different layout for your carbon nanotube. And this, as we said, affects the conductivity and strength. So the vector can be thought of as residing upon an infinite graphene sheet, and this chiral vector describes how to roll up the sheet to make that tube. And T, as we said, is denoting the axis of the tube. So this vector can affect, as we said, the properties of carbon nanotubes, and specifically, depending on the vector, the electrical conductivity, which is of more importance to us, will change. An equal number of unit vectors will result in a metallic property for the tube, so it will be a very good conductor, whereas if this difference between N and M, if it's a multiple of 3, in that case it will end up with a semi-conducting nanotube. The nanotubes will, with different structures, can then be networked. You can use them together, like the metallic nanotubes and the semiconductor nanotubes together to form a network and produce parts for circuits and for standalone networks. You can also, by changing the structure of the nanotube at a specific position, for example, by bending the nanotube, you can change the conductive properties. Because this layout affects how, for example, the holes propagate in the case of semiconductors. And from one end of the tube to the other can be changed, which will actually correspond to a resistor in electrical circuits. And the fact that a slight change in the winding of these hexagons has some important effects, like turning it from a metal to a large gap semiconductor, has some very important implications. And these can be useful for using these nanotubes to construct actually semiconductor devices, or networks of carbon nanotubes can be used. Unfortunately, up to 1/3 of the tubes appear to be metallic. As I said, you can synthesize them as human beings, but you don't produce them, of course, one by one. So when you try to produce them, most of them turns out to be metallic, so you have to live with that. Carbon nanotubes also have excellent thermal conducting properties, with an estimated thermal capacity of up to 6,000 watts per meter Kelvin, which is much better than the standard copper wire. So they're really very good conductors of heat. So now suddenly, I can think about a different way of communications. How about transmitting heat? Remember, when you try to transmit information, you have a signal of something, molecular wave, electricity, electromagnetic field, whatever, and you try to encode the information on some feature of the signal. How about using the heat for transferring the information? If I can efficiently transfer heat, can I use it for communications? That could also be an alternative. So this opens up yet more opportunities for the use of carbon nanotubes, for example, for heat transfer. Another property of the multivolt carbon nanotubes is the telescopic effect of an inner core of a nanotube inside the other. So if you have two nanotubes, two concentric nanotubes, now you can shift one inside the other. And what's good about it is, now if you have two structures, one going in and out, the other in a telescopic manner, typically you have friction, right? This is almost frictionless. So it's really efficient. Now, of course, to be able to do that, you should be able to hold the outer tube so that you can shift the other one inside that. Otherwise, they could be moving together. So to have that telescopic effect, you should fix one of them. There are things like buckyballs that are consider the nanotube as a tube and assume that there is something like a ball attached to the top of it, which could be used as a handle to hold the outer tube so that the other one can move inside. The second tube, the inner tube, can move inside the outer tube while the outer tube is fixed by means of that buckyball. So that's also yet another way of using it. So this might result in yet a different way of communications. This time you can send the information by just pushing in the destination, since you have that nanotube. So that's yet another way of transmitting information. So that could be, for example, another use could be through the vibration of the carbon nanotubes. These carbon nanotubes have been shown to be capable of oscillating in the terahertz range. And in addition, people are looking at these things in more detail. Recently, it has been shown that the ability to construct a functional radio receiver from a single carbon nanotube operating in the 40 to 400 megahertz has been possible. This is nice range. This is something we can use with our regular wireless devices. This is important because this radio receiver is operating at the frequency band I can use in my daily radio devices, and the radio devices I have at the moment. So that means it can work as a gateway between the human scale and the nanoscale. If these carbon nanotubes operated at a frequency that was not feasible for us at the human scale, I would not be able to communicate between these two devices because they're not working in the same frequency. The nice thing is their frequency bands overlap. This is very important. That's what makes it possible. So the radio can now receive and demodulate both AM and FM signals you use in your radios. So this opens amazing possibilities for us, not only for using radio communications inside the nano networks, but also using wireless radio communications to connect multiple nano networks and the traditional networks we have. The carbon nanotubes show great potential for many different uses, as we have seen, and their amazing properties, including their conductivity, starting from conductivity all the way to strength, make them a prime resource for realizing the nanonetworks and implementing computer chips. But also, not only for implementing computer chips, we can use them for other purposes, as we have discussed. Research typically continues towards additional possibilities on how to exploit these features in different ways for nanoscale structures in nanonetworks. For a list of some of the current tools for simulating carbon nanotubes and nanocircuitry, please have a look at Appendix C in your textbook. You will see some references there, useful references. And more research is still required in experimentation, in how to do manufacturing. That's very important. Now, first, the experimentation is done in the lab environment. But once you're able to produce something in the lab environment, you should find an efficient way of producing those things in mass production manners so that it becomes cheap and a usable technology. Otherwise, it would be useless. Modeling and simulating these properties and information theoretic research for the new communication paradigms that are being produced. So, any questions up to this point? These are some of the features of carbon nanotubes, but I hope it was encouraging on how useful these devices could be. But now we can talk about the relationship between these carbon nanotubes and the microtubular networks we had discussed in chapter 2. So remember, the microtubules were typically working as the railways inside the cell to transport information, which was enclosed in messenger molecules in the vesicles. Or also those microtubules were useful for example, transporting organelles inside the cell. You can pull the organelles actually around also. That's also possible. In a sense, the microtubules are the cell's own version of nanotube network. Not carbon nanotube, but nanotube. Because remember, the microtubules actually were like tubes. Remember those alpha and beta tubulin structure of that microtubules? It is actually like a nanotube. Also, it's at the nanoscale. So it's very similar in that sense. But there are also some differences. Okay? So there's a clear similarity in their structure. As we said, they're both hollow and thin-walled tubes, again with a very high aspect ratio. And they're very efficient for bearing loads, for carrying things around. The microtubules are cytoskeletal biopolymers that play a critical role in all phases of the cell's life cycle, for transporting information, for transporting useful chemicals into the cell, for transporting hazardous chemicals, the waste products, out of the cell, for many different purposes they could be used. They're also useful, for example, in the split of the cell to two pieces when the cell is trying to reproduce. It is very useful when the cell is trying to move, for example. With the help of the cytoskeletal, the cell is able to form limbs that extend either for movement or for grabbing some useful molecules from some neighborhood. The microtubule, or the cytoskeleton formed by the microtubule, helps the cell membrane to hold tight, forming the cell's shape. So it is useful in many respects. So they provide mechanical stability for the cell, including holding its shape during cell migrations, and also provide tracks for intracellular transport of these, for example, molecular motors. The microtubules are 100 times stiffer than the other cellular components in the cell and have a high degree of resilience. Carbon nanotubes are also suggested to be the closest to non-biological counterparts of the microtubules. And the carbon nanotubes are extremely stiff with a Young's modulus which is 5 times higher than that of steel. Okay. The microtubules are also highly resilient, but while the chemical composition of the microtubules is comprised of proteins and non-covalent bonds, it is different. That's one of the differences with carbon nanotubes. In the case of carbon nanotubes, this time we have carbon and covalent bonds, as opposed to the non-covalent bonds here. But their mechanical behaviors are quite similar. Both microtubules and the carbon nanotubes are spontaneously assembled into bundles. That could also be done. The microtubules and the carbon nanotubes share similar electrical properties. The flow of the current through the microtubules and carbon nanotubes is a different process. In the microtubules, you have an ion channel for transporting electricity, whereas in the case of carbon nanotubes, you have semiconducting properties or the metallic properties for the transfer of electricity. The current flow through microtubules was measured you can find the reference here, to be approximately 9 nanosiemens at a rate of approximately 1 meter per second, which is quite fast for nanoscale networks, and it exhibits an amplification effect. So we could even draw the analogy further to say that both microtubules and the carbon nanotubes can act as transistors because they are able to amplify the incoming signal. Suda and Michael Moore have alluded to the fact that electric fields may be used to control the positioning of microtubules. We'll briefly discuss about this later on also. But you can change the behavior or the positioning of the microtubules by playing with the electric field. And both microtubules and the carbon nanotubes are impacted by magnetic fields, and free-floating microtubules can be steered also by a magnetic field. And microtubules naturally self-assemble, while the controlled self-assembly of carbon nanotubes is possible by coating with amino acids. One significant difference between microtubules and carbon nanotubes is that the microtubules are more dynamic than the carbon nanotube networks. The microtubules, as you will remember, they're formed by centrosome, but while you're assembling the microtubules from one end, at the other end it keeps on dissolving. So remember, the microtubular network was very dynamic. That's not the case for carbon nanotubes. They're more durable. So that's one major difference in between.
Do they have a lifetime, carbon nanotubes?
Of course, everything has a lifetime, but it's pretty long. I cannot tell you exact values, but it's pretty long compared to microtubules. So microtubules are switching between phases of assembly and disassembly in time. So they constantly grow and shrink in size. Also, microtubules commonly appear in a band shape. That's also an important feature due to the strong cytoskeletal forces. They're not in straight lines, whereas in the case of carbon nanotubes, they're mostly cylindrical. That's also an important difference in between. Remember, when we were discussing the microtubules in chapter 2, we talked about some persistence length. So the persistence length was actually the rate at which the tangents taken along each segment of the microtubule become decorrelated from one another. That means consider one microtubule, take the tangents along that microtubule. How fast are these tangents becoming decorrelated? If they're close to each other, if two neighbor tangents are close to each other, then you can say it's almost straight microtubular section there. Whereas at some sections, the tangents will be quite different. They will be decorrelated. Where you can say that is the end of one microtubular section, another section starts. Okay? Which are decorrelated from each other. So that persistence length quantifies the degree of bending of the microtubule. If R of S is a point on a segment of the microtubule, say S, then let U of S be the unit tangent vector at that point. So U of S can be defined as the derivative of R with respect to S. So the orientation of the unit tangent vectors for the segments 0 and segment S can be quantified by the inner product here, where iota is the persistence length. For longer persistence lengths, which means more straight microtubules, or for shorter tubes, the microtubules will be straighter when you're comparing them. It will look relatively more straighter. For longer tubes and shorter persistence lengths, the impact of decorrelation along the chain tangent becomes more significant, and we can approximate the curved microtubules as a sequence of actually shorter segments, smaller random chains that are like appended to each other end to end. Any questions? The last things were something we already discussed in chapter 2. Now we can talk about the motility, that's a movement assays for these carbon nanotubes used in junction with microtubules. So carbon nanotubes have been associated with the biological applications for some time, and they have been used to, for example, mimic neurons carrying and delivering drugs into cells, and they have been also coupled with both DNA and molecular motors. And there is therefore some cross between the inorganic and organic nanoscale components. And this is important for the advancement of nanonetworks and nanotechnology in general. So motility assays is a means where you have this interaction between the carbon nanotubes and the molecular structures, typically microtubules. So the motility assays have been engineered in two distinct geometries. One is the bead assays, and the other one is the gliding assays. The bead assay is one in which you have the microtubules or the filaments that are fixed to the surface, and the molecular motors are moving on these. So once again, the tubular structure, microtubular structure is fixed. The molecules are running on these microtubules. Whereas in the case of gliding assays, it's just the inverse. This time, the tails of the motors are fixed, not the heads, of course, because that's the part that is touching the microtubule. So that part has to be free. But the tails or the neck of the molecular motors are fixed, and then you release the microtubules on these motors. So when they try to move, since they're fixed from the neck or from the tail, they are actually, while trying to go this way, they're actually pushing the microtubule or the filament this way. So therefore, this time we have fixed tails for the motors, and the heads of the motors are now carrying the microtubules across the surface. So the figure here explains this in more detail. The order of these in the figure are just the inverse, actually. In the case of bead assays, you have a siliconized glass on which the microtubule is fixed, and then you have the molecular motors, kinesin or dynein, whatever, running on top of these microtubules, just walking as we used to use them, as we used to discuss in chapter 2. Okay, so the motors are running on top of the microtubules. The gliding geometry is different, or in gliding assays, what happens is now the motors are fixed, and you put a microtubule. The molecular motors try to walk, but while they're walking, actually they're not moving at all by themselves, but they're pushing the microtubule in the other direction. So you can benefit from this idea, as we will see now. For example, in reference 6, the carbon nanotubes are oxidized using a sulfuric and nitric acid mixture. And then these oxidized nanotubes are mixed with streptavidin proteins, and they're incubated to form the streptavidin-coated carbon nanotubes. This streptavidin is actually a protein that comes from the bacterium with this name. I won't try to pronounce it. So this streptavidin protein is widely used in molecular biology because it has an extraordinarily strong affinity with the molecule biotin. Actually, if you look at the affinity between biotin and streptavidin, the dissociation coefficient, constant of the biotin-streptavidin complex is on the order of 10 to the minus 15, which means they don't want to dissociate. They want to keep together. That's why, since this number is very small, they have higher affinity. So it's ranking among one of the strongest non-covalent interactions known in nature. Okay? So, streptavidin forms a strong linkage, therefore, with the microtubules that have been functionalized with biotin, covered with biotin. And the streptavidin-coated nanotubes are now able to bind to microtubules that contain biotin tubulins, and the carbon nanotubes can move on a gliding assay. So this technique is typically used to attach the nanotubes to the microtubules, and the molecular motors, which are now fixed on the plate, fixed at specific positions along gold-plated terminals, and the molecular motors now are carrying these microtubules, which have the carbon nanotubes attached to them over the area. This is just like crowd surfing in rock concerts. Okay? Actually, in the case of crowd surfing, the audience is fixed with their feet on the ground, but their arms are now moving, carrying the rock singer over the area. Now, if you can place these people also, and these spectators who are voluntary to carry this person in a specific pattern in the audience, you can take the rock singer from the stage for crowd surfing and throw him out, okay, as you wish. So the molecular motors are therefore shuttling the microtubules with their cargo, which is the nanotube, across the terminals, from one gold-plated terminal to the other, for example. ATP is typically serving as the fuel for this operation, because you're consuming energy for doing this. So what you can do is, remember, for the binding between the molecular motor heads and the microtubules, in every step we were using 1 ATP. Now, if you can spread this fuel, like putting some spice on your food, forming a path, the microtubule will not be able to have this moving effect with the molecular motors that do not have that fuel. They can only have this reaction with the molecular motors around which you have ATP. So what you can do is you can fill this area with these fixed molecular motors and spread the ATP as you wish in a pattern that you wish, and just leave the microtubule from there, and it will follow the path over which you have spread that ATP. So you can also control it this way. So this is actually explaining what we discussed before. So on a glass surface, you have the molecular motors like the kinesin attached here, fixed there, and on top of it you put the microtubule, which is covered with biotin, and on top of it you put the nanotube, which is covered with streptavidin. Not the whole thing is covered, of course, but these are the attachment points. Since the streptavidin forms very strong bonds with the biotin, the nanotube will be attached to the microtubule, which is carried around on top of the molecular motors. The movement direction and the speed of the carbon nanotubes can be controlled in several ways. one thing is, as we discussed before, the electric field. Negatively charged microtubules move faster in the presence of assisting fields, and they can be completely stalled in the presence of opposing fields. So you're able to control the speed and the movement of the microtubules, if you wish, using electric fields. So this is, pay attention, the electric field is something you can control at the human scale. So you can control the movement at the human scale, control in the nanoscale movement using some human scale tools. Another one is light, specifically ultraviolet light exposure. What you do is, this time you put caged ATP, that is ATP which is somehow deactivated, it's caged, and in order to uncage it, make it active, what you do is you provide ultraviolet light. Okay, so you first have this caged ATP all over the surface, then by exposing a path to ultraviolet light, you uncage the ATP there. So the caged ATP is not active, so it cannot help in this hydrolysis, in the molecular motor movement. But the uncaged ATP now helps in this movement. So again, the microtubule follows the path you're marking with the light. That could be another way of controlling the motion. You can have physical tracks. You can form tracks on the surface of the medium that can guide the motion. So, for example, a microtubule could be going towards the obstacle. You can make it turn, or you can make it escape also. By playing with the ATP concentration, you can control it. Like variable ATP concentration in the medium will guide the motion, and controlled release of ATP will determine the speed. So you can switch on or off the movement. So you can physically guide how it will work. By making use of chemicals also, you can control the motion, like using molecules that mimic microtubules or inhibiting the movement of the kinesins. You can control the movement. Thermoreactivity is yet another method. So embedding the kinesin molecules within a thin layer of thermoresponsive polymer, PNPM, which stays in contact state at some specific temperature, you can control the movement. And by changing the temperature, you can control whether it will move or not, or you can attach or detach it to the plate, for example. The nanotubes can also be functionalized in different ways to enable drug delivery. This is typically used for cancer treatment, for example. So they can be oxidized using acids, resulting in the reduction of their length to aid their ability to disperse within some aqueous solution. Also, the chemical processing can be used to change the chemical structure of the carbon nanotube external walls and tips, making them soluble in water. Thus, you get functionalized carbon nanotubes, CNTs, that can be coupled to a variety of other biological molecules, including peptides, proteins, nucleic acids, and other therapeutic agents that you want to send to the cells. The fluorescent markers attached to these FCNTs can be used to track their ability to easily penetrate the cell wall. So you understand that it really goes through the cell wall or not by making use of the fluorescent markers. The fluorescent markers are typically used for several purposes. The most important thing is, under microscope, you can see these markers. So you understand where the marked elements are going to. So you understand, for example, that it enters the cell. Evidence given in 12 shows that the macrophage cells could ingest significant amount of nanotubes without apparent toxic effects. Translation says, carbon nanotubes can penetrate cell membrane without triggering the self-defense mechanisms of the cell. This is very important. Okay? Typically, when you try to tamper with the cell, if it thinks that the integrity of the cell will be broken, the cell typically commits suicide. That's not the case when carbon nanotubes are used. So in terms of drug delivery, one example is the transport and uptake of DNA by a cell. So injecting DNA alone as a plasmid has difficulty in reaching the cell nucleus, but when you use it together with carbon nanotubes, you can do this. So when DNA is attached to FCNT, the gene expression levels increase between 5 to 10 times. Okay?
Saying attached, do we mean putting the DNA inside the carbon nanotube?
No, not inside, but together with it. Attaching them. Yeah. Okay. Typically, carbon nanotubes are very small, so it's difficult to insert something inside.
I always think about, when you say tube, microtube or carbon nanotube, I always think about putting inside and transporting in that.
No, not necessarily. That could also be done, but not necessarily. There's one thing we should note. Now, in these slides, it appears like carbon nanotubes are very good for health purposes. Now, this is an ongoing discussion. Carbon nanotubes can be used in, as we discussed here, in drug delivery, so in that sense they're good. But there are also people arguing that the carbon nanotubes, just like asbestos, are harmful, especially for the lung cells. So being subject to carbon nanotubes in an uncontrolled manner, that could also be very hazardous. So this is an ongoing debate. I'm not able to comment on which one is true, but I just want to inform you that it is still under discussion whether carbon nanotubes should be used in health applications or not. It could be, they could be used, for example, in drug delivery, but for example, in a controlled manner. Okay? The problem mostly arises from the fact that these things can pass through the cell membrane. So we should be able to control how that works. So yet another use could be using the carbon nanotubes as field effect transistors. So to construct a nanoscale network, we should first understand the medium in which this network is operating. So we have previously discussed the chiral vector, and the chiral angle is the angle of that vector with respect to the A1 vector, remember, we discussed. They describe the atomic structure of the tube and its properties. In the case of field effect transistors, then the important thing is the electrical conductivity. So the angle indicates to what degree the lattice is twisted, how you roll it, in other words. And there are different names depending on how you roll it. Like if the chiral vector is n by n, in other words, m is equal to n, in that case it's called armchair. If m is equal to 0, it's called zigzag. And if m is half of n, or in other words, 2n by n, in that case it's called chiral. The nice thing about this structure is that depending on how you roll it, the nanotube can have different conductive properties. The lattice of these carbons has the property that it can backscatter the electrons, moving at the Fermi energy, which gives the nanotube the property of a semiconductor. Okay? If the electrons move along the y-axis at 60, 120, or 180 or 240 degrees to the y-axis of the lattice, then the backscattering is suppressed. So depending on how the lattice is arranged, as we said, we can adjust whether it will be a metallic or a semi-conducting carbon nanotube. More amazingly, the semi-conducting nanotubes can be made into transistors if you apply either a negative charge, which will introduce holes into the system, into the carbon nanotube, that will enable conductance. But then, if you cut out that negative charge and apply positive charge, this time it will deplete the holes and reduce the conductance, which means you can control the conductivity, which was the idea with semiconductors. So these properties explain why the carbon nanotubes are an active research in nanonetworks and circuits, and more work has been conducted also to see these, how these perform. The molecular dynamic simulation to test the carbon nanostructure for buckling and deformation. You have the tube created by rolling in some direction as you wish, according to the chiral vector. And then you try to bend it. So there is a point up to which it can resist, but then there is, you go beyond that limit. So that is buckling, and then beyond buckling, then you have deformation of the carbon nanotube. So there's a simulation, dynamic simulation for that. So trying to find different buckling and deformation for tubes of different diameters and differing energies. An interesting result from this paper is that the simulation seems to indicate the tubular cross-section can deform as much as 20% before that buckling occurs. So it has some resistance to that force up to 20%. And after that, you have deformation. And a new study further investigates this, looks at only the mechanical properties like buckling, but also looks at energetic and vibrational properties of the single volt nanotubes. Specifically, the nanotubes in question are the armchair layout n by n, and zigzag n by 0, and chiral 2n by n. And the study was able to identify the stability domains for each one of these circular and collapsed cross-section structures of the three types of single volt nanotubes, these three different types. And using a theoretical model and simulation, the study identifies the different radii points for which different radii become more energetically stable compared to the others. So with which radius you should be working. And another study also gives the simulations for the vibration modes and the frequencies of these single volt carbon nanotubes, specifically for the armchair, and finding that the simulation matches previous experimental frequencies. So all these will typically help to develop a model for information transformation based on vibrations in this case. And there are also other studies working on stiffness and also examining vibration. And large-scale simulations are also being conducted. So if you're interested in these, please refer to the book, to the corresponding references, and you can get more information there. And as I said, go to Appendix C for more detail about these simulators. I'm not going to, of course, those specific simulators. Thermal conductivity is yet another area where we can make use of another feature of carbon nanotubes that can be used for communications. So if the nanotubes are going to be used for nano devices, then managing the thermal effects will be important to carry information, for example, from one side, one nanomachine to the other. So the exact thermal conductivity of the carbon nanotubes is yet to be fully understood. And as we said, the conductivity can go around 6,000 or 6,600 watts per meter Kelvin. To overcome the difficulty in performing the thermal conduction measurements due to the problems with synthesizing and aligning high quality carbon nanotubes, a molecular simulation model again has been developed in 24, and according to this model, the advantage of not requiring high numbers of parameters deduced from the experiments. This model does not have so many parameters as the Boltzmann equation requires actually. And that model corresponds well with the previous results, converging to approximately 300 watts per meter Kelvin. That should be uppercase, sorry. And the review of the computational simulation tools that can be used to conduct modeling studies is also provided in 24. Another tool that could be examined is on the vibration and flow of heat problems. This is nanoheat flow. In this one, the molecular dynamic simulation of time evolution of kinetic and heat energy is provided. So that would be all for, this week. That's where we stop. Next week, as I said, you will have the exam. You're not responsible for this chapter in the exam. The following week, actually, I will not continue with where I stopped now. The following week, we will have Tugcu and Birkan, my postdoc people, who will be discussing the works we have been doing in our lab on molecular communications. They will go into details of, for example, their findings in diffusion. Specifically, they will be talking about Brownian motion. We will see that actually the Brownian motion has already been studied in detail, and there are some very important findings and things that will actually simplify your studies if you plan to work in this field. And most of those studies are there in the literature, and people working in this field are not aware of it. So that will be quite helpful to you. So I believe you will enjoy, or at least benefit, from their presentation. And after that, we will continue from where we were left in chapter 4 and complete this discussion. We'll be talking about that single carbon nanotube radio in more detail. And we will also be discussing the deployment of nanotubes to create a network. So that's all for this week, and hope to see you all next week in the exam.